So today I'm going to tell you about some very new technology. I think it can revolutionize the way we type people for blood groups, and hopefully I can convince you of that. So over 100 million units of blood are collected each year worldwide. These are truly life-saving for people who get them for cancer, in trauma, pregnancy, sickle cell patients, stem cell. However, transfusion is not without complications. Between 3 to 30% of people will form an antibody against blood group differences between the recipient and the donor. These antibodies actually can lead to transfusion reactions, the most devastating being a hemolytic transfusion reaction that can be fatal. And in fact, after the early transfusions led to many deaths, transfusion was banned for hundreds of years until we understood the basis of blood groups in the early 1900s. In addition, these antibodies can lead to hemolytic disease of the newborn fetus, again, with devastating consequences. And once these antibodies form, there's really nothing you can do. They're there for life. As a result, we match people up front for ABO and RHD. These are the blood groups you've heard of. And the reason we do that is everyone has a preformed antibody against foreign differences for ABO, and RHD was the #1 cause of hemolytic disease of the newborn fetus. So it made economic sense to try to go after these first. And the way we do that really hasn't materially changed since the 1960s. And we take patient red cells, we isolate them, and we incubate them with antibody reagents that are specific for that particular antigen. Now the challenge here is you need to make an antibody reagent for every antigen you want to type. And that's not doable at large scale. So we have antibody reagents for a certain number, but not for everything. And the readout is simply just agglutination, do the red cells clump or not. This technology is still not even very good at getting 100% coverage for even just ABO and RHD. There are a number of very interesting ABO subgroups, some of which important really clinically for transplantation, which is an emerging market for getting better ABO typing. As well as in RHD, there's some partial and unusual weak D forms that have implications for pregnancy and whether or not you give ROGAM. And these can't really be easily teased out with conventional serology. So again, a very important thing to figure out. There are 300 other blood groups. They are split among blood group systems, and here are some of the names of those. And so why don't we're now going to dig in a little bit and sort of explain to you why have you never heard of these other blood groups and why should you and why should we care. So here on one side is an example of all of the antigens in one particular individual. And you can see in green, the A and the D antigens are highlighted somewhere in that long list of many antigens, all of which we'd really care about if we could know about them. Now, routine serologic typing is pretty inexpensive, and it can type for those two antigens, as I just mentioned. We can do extended serologic typing, and we do this for chronically transfused individuals like a sickle cell patient, someone who's going to get 10, 20 units of red cells a year, and they're going to start making antibodies eventually. So we're going to want to try to prevent that. So it's now clinical standard of practice to do extended serologic typing on them. But this is very expensive and very labor intensive. This is manual typing. This is technologists with tubes in their hands shaking them. There aren't really machines that can do this level of typing. The ABO and RH have automated platforms because they're sort of just one-offs. But to do this larger scale typing is not really feasible for even more than just a handful of antigens here highlighted in green. In addition, commercial reagents aren't available for the vast majority of antigens. The only source of antibodies would be a patient that had made that antibody, and you have isolated and identified that. And that is not a scalable, commercializable approach to typing. And it's very, very expensive. We're talking $500 to $1,000 to start typing these additional antigens with labor. And some of the antigen reagents, especially the ones that are rare, are very, very expensive to buy. As a result, people have started looking into molecular methods, DNA-based methods to type for antigens. This has really taken hold in the HLA arena where almost virtually all typing is now DNA-based and not serologically based. One way to get about that, a cheap way, is to use single nucleotide polymorphisms. That is, if you know a particular DNA change and how it relates to a phenotypic antigen change, you can look for those particular DNA changes and then infer the antigen type. Even here, there are some chips on the market. They still have limited coverage of antigens they type for. Some of the reasons here are the chips that are on the market are a little bit older technologies. So they are very constrained in the amount of SNPs they can look at. In addition, some of the antigenic changes we're interested in are not really amenable to SNP chips. They have large structural rearrangements, as well as some very hard to establish relationships between nucleotide positions within a particular gene. So they're not really amenable to getting at the large majority of antigenic changes either. But they are cheaper. You can scale them. and you can get and people are now doing this for large-scale screening of donors for particular antigens that are covered by these chips. So if you could come up with a technology that could get the other antigens, there's already markets and people there that would be interested in using these, even at a slightly higher cost than conventional serology. So that's where we decided to try to establish whether or not next-generation sequencing, and in particular, we focus on whole genome sequencing initially, since we get the largest amount of data. And here you can see now all the antigens are green. And in fact, this is a true antigen repertoire in one individual that I manually typed a few years ago. We used standard whole genome sequence, and we got the variant calls at the end, and I went manually for a few months and tried to annotate all of those antigenic changes. We had another 100 in our trial that we were going to do, so obviously that would take forever. And so we created bloodTyper, which is an automated software that can do that analysis, and it do it much better and quicker than I was able to do it, actually. And so to date, we've done 300 whole genomes through that platform. And now these genomes are really unique genomes. These all have extended serologic phenotyping and SNP phenotyping. So these are highly characterized genomes that allowed us to establish a really good concordance estimate. And in the first 300 genomes, we did several rounds of blinded testing. So on blinded testing, the current algorithm is at 99.9% concordance with conventional typing. And to put that into perspective, in one of our earlier rounds of blinding, we were able to follow up and get repeat samples for all discordances and found that the conventional typing had about a 0.1% discordance rate as well. So this is nearly sort of equivalent to conventional typing, but you're able to get all the additional antigens. Now, obviously, cost is an important consideration here. A whole genome is somewhat expensive still. However, that cost will go down over time, whereas the cost of doing extended serology won't go down. In fact, the cost of doing extended serology will likely go up as SNP-based and other molecular methods enter into the market. There'll be less and less vendors making these serology reagents. And at some point, like in HLA, you won't even be able to buy commercial reagents for some of the antigens, most likely, that you could even today. But one of the big markets here would be existing genomes. You look to the 100,000 Genomes project, the All of Us sequencing project here in the US. There are now some large-scale initiatives to sequence populations of individuals. This is an algorithm that could then operate on those populations at a very low cost and increase your return on investment on sequencing. And we think that if you're a country and you're going to sequence your population, this would definitely be an analysis you'd be willing to pay a few dollars to run on your genomes, since it'll help you find better donors in your network and hopefully reduce transfusion complications and decrease the overall cost of transfusions while making them better. In addition, you can also look at whole genomes. The whole exomes, these are slightly cheaper. This is something certainly you might do if you have a very complex case at a reference lab today, even. The cost of that would be equivalent to just doing some extended phenotyping. So it would be very, very within the cost structure already. In addition, you can constrain that and do more scalable assays where you just look at a subset of genes or even just one or two genes. And in particular, we've already done a proof of concept on the ABO gene here, where in stem cell transplantation, the first sample is actually a buccal swab in which you isolate DNA and do HLA typing. You don't get a blood sample. And we'd like to know the blood type up front on those donors, since we'd like to match for ABO as well, rather than having to call them all back in and get blood samples. So as a result, we do a lot of mismatched ABO transplants today, which lead to morbidity and mortality outcome data. So with a DNA-based assay that we've been developing on this targeted next-gen ABO typing assay, you could do upfront from buccal swabs ABO typing at a very low cost, since you could scale this across hundreds of samples in a run. So now that we've developed that technology, we are looking to commercialize it and license bloodTyper out for use. As I mentioned, sort of some of the use case scenarios would be sort of large-scale sequencing as well as targeted assays. Similar to, I think, a lot of other people here, there's still some development work to be done on the software to make it more easy to use by end users, as well as to distribute some of that software into the cloud, especially, for example, to be allowed the targeted ABO assay to be called just from your sequencer using a cloud platform would really simplify the integration for an end-use lab, like an HLA lab that would like to use that alongside their HLA typing. In addition, there are many other antigens out there that we weren't able to capture in the extended phenotyping in terms of concordance data. We'd like to test for those. That requires finding unusual samples, which we have collaborators, and at the Brigham we have some of these samples. We just need to sequence them on a whole genome or targeted assays in order to prove that concordance. And we'd like to develop some more specific assays. With that, I thank you for listening, and I'll be outside if anyone has more questions.